Thank you everyone for coming to this panel session at Neuromatch 4.0. I am Grace Lindsay. I'm a postdoctoral research fellow at the Gatsby Computational Neuroscience Unit at the Sainsbury Wellcome Center. I'm also the author of the book Models of the Mind, How Physics, Engineering, and Mathematics Have Shaped Our Understanding of the Brain. And this panel is going to be all about models in computational neuroscience, in the field of neuroscience, the role that they play, where they fit into, and how we go about developing them. And so for that, we have a very, well, we had 4 people on our panel for a minute there. I believe we will have 4. We have 4 very esteemed people who have worked in the field of modeling and neuroscience in a huge, diverse array of topics. And we're hoping to get some insights about, what has made modeling fruitful in the field from these panelists, and where we think modeling can be going in the future, and how it can help further advance the study of the brain and neural systems. Before we get started, I want to remind people that they can put their questions to the panelists in the Ask a Question tab at the bottom of the page. I do want to save some time at the end to get to some audience questions, so do put your questions there as you have them, and then also you can go there and upvote questions that you think are particularly interesting and you want to get asked in the limited time that we'll have at the end. So that being said, I think that's all the administrative details out of the way. So I want to introduce our panelists for today. The first panelist we have is Eve Martyr, who is the Victor and Gwendolyn Beinfield Professor of Neuroscience at Brandeis University. Randall O'Reilly is the Professor of Psychology and Computer Science at the Center for Neuroscience at the University of California, Davis. Alessandro is full professor at the International School of Advanced Studies in Italy, and Christoph Koch is the chief scientist and president of the Allen Institute for Brain Science in Seattle. So I've kept those intros short because actually the first question I want to pose to the panelists is for you guys to tell us a little bit of how you came to this intersection of computational modeling and the study of the brain or neural systems. So what drew you to this kind of work, and particularly, the niche that you have worked in for your career. So if you guys can give us a little bit of your biography in that sense, I think that'd be really interesting to show at first the diversity of models represented here, and also the different motivations that people have for using them. Maybe we can start with Eve. You're first on the screen for me.
So thank you. I'll try and keep this short. I'm a trained biologist, or I was trained as a biologist, and I was studying the dynamics of neuronal neuromodulation in a small crustacean nervous system, the systematic nervous system. And this comes from lobsters and crabs. And about the late 80s, we had already figured out that the wiring diagram was insufficient to really explain dynamics. And we had already figured out that most normal mortals, such as I was, find it difficult to reason about the dynamics of more than 2 cells at the same time. So I was sort of driven to some of my theory friends to try and get another tool to try and understand dynamics of even small circuits, because it was clear we couldn't just reason with our own sort of lack of understanding of dynamical systems with our brains alone. I mean, I can tell just so stories. And so in the late 1980s, I started working with people coming out of physics and dynamical systems mathematics. And my earliest collaborators were Larry Abbott, Nancy Capel, and Irv Epstein. Irv was a dynamical systems chemist. Nancy was an applied mathematician, and Larry was a physicist. And those 3 people were extremely important in setting us on this stage.
Great. Randall, you want to go next?
Sure, yeah. My story actually starts in high school, at least. I was really into programming computers, and I had this book, I don't know where I got it, but it was about AI. And I read that book and I said, oh, I'm gonna program an AI, and tried to do that. And I failed miserably, of course. And I actually was planning to go into physics, and went to college and enrolled in physics, and then sort of went to the psychology library one day and sort of said, hey, wait a second, look at all this interesting stuff that people are actually doing. And I think it was after I had met with my advisor who said I was going to spend, the next 5 years doing all this really boring physics. And I was like, I only want to do the interesting physics, and then I could do this interesting neuroscience and psychology stuff right away. So anyway, I had this intersection of those kinds of elements in my background of really, enjoying programming and thinking about how to actually sort of implement something on a computer, also having the kind of math physics type background, and then really just being really interested in neuroscience. And then, I sort of went through college and then found, Jay McClelland and went to grad school with him, and sort of have always basically been doing more or less the same thing, this kind of computational models of neuroscience, basically, psychology.
Alessandro?
So I wanted to be a theoretical physicist, and I went to Jerusalem for my PhD after the
Alexander, can you move your camera
Yes.
over? Your face is cut off.
Yes, like
That's
this.
better.
Yes. So I moved to Jerusalem for my PhD after getting the Italian degree in physics, and I wanted to do fundamental physics, but it was beautiful, but there was a kind of an atmosphere of frustration that the important results in what was then renascent string theory were the privilege of very few very mathematically talented individuals. And at the same time, in Jerusalem, just where I landed by chance, there was this new thing that was very exciting of the physicists that had begun to study models of associative memory with the Opfield model. And then those three key people in Jerusalem, Daniel Amit, Hannah Gutfreund, and Chaim Sompolinsky, had understood early on that this thing could be just a game that would pass in a few years, or it could be something more serious. But then they had to really learn about the brain, about which they knew nothing. And so they organized something in 87, 88, which was an interdisciplinary year, where everybody in Jerusalem who had something to say about the brain, psychologists, neurophysiologists, even philosophers, computer scientists, presented their stuff. And basically the physicists, the professors, and the students were there to learn. And it was really a very nice experience. And it was a bit the beginning of that particular branch of computational neuroscience done by physicists. So I never really decided to join it. I was just swept in the current.
Thanks. Christoph?
Yeah, thanks, Grace. By the way, I'm not president anymore. I stepped down during the pandemic. I'm not chief scientist. So the brain generates behavior. The brain exudes, it's also the substrate of consciousness. The brain may be a computer, but whatever else the brain is also a physical system subject to the same laws as any other piece of furniture. So it ought to be studied, among others, also as a physical system. So in my case, I'm very interested in action potentials, synaptic potentials, local field EEG. So one can study the system at that physical level. Where do the current flow? And unlike if you look at, for example, genes, there are conservation laws that are obeyed, right? All the current that flows into a particular piece has to equal all the current that flows out. So it has something very concrete, and ultimately biology and neurobiology and behavior, of course, has to arise out of that physical system, which is why I think it is essential to also treat it as such and to model as such. Ultimately, if you want to understand a piece of brain like cortex, we have to also understand it at the physical levels. Where the currents, how do they influence, what are the causal actors that drive the various operations that leads, let's say, from photons on the retina ultimately to visual behavior and visual perception.
Great. So I think we have touched on in your answers kind of the different topic areas that you all apply modeling to, and it really ranges from, Christoph, as you were saying, really studying the cell as a physical system, individual neuron models or neural circuit models, but then up to work done, by Randall and Alessandro to some extent on more kind of AI or artificial neural networks about how whole systems learn and implement memory, and all these different things. And of course, modeling can be applied anywhere in neuroscience because it is just kind of a way of understanding a system. And as Eve said, when you realize that your brain is too feeble to keep track of everything, you need to turn to modeling. So I want to, despite the diversity in areas of neuroscience that modeling can and has been applied, I want to try to kind of get at maybe some universals to what has made a research topic or question particularly ripe for modeling. So in your past experience, has there been something that you've really thought about, like, oh, there's so much data here and people haven't pieced it together with a model yet, so I should come in and do that? Or maybe the opposite, there's no data here, we need a framework to start, building some kind of understanding. Have there been traits of a question or kind of a research environment that have made you think that, yeah, we should definitely start doing some modeling here in your past work? Anyone can.
Well, I mean, there's modeling at different levels of resolution. There's something like the Hopfield model, right? That has been enormously influential, but that's of course at a very abstract level. And then there's model like of the sort that Eve or I do, where you try to measure channel densities and where are they located on the dendrite, that are very, very physical. And then there's everything in between. So I think in any field one can have models. All models are wrong. Some models are useful, and if they're useful, they give rise to new interpretation, and they let us understand a little bit about our experiment, and then derive or devise the next experiment. And I think that's their main function.
So I would like to say that I always tell young theorists that the best way to approach experimentalists is to find a really smart experimentalist and ask them what he or she doesn't understand. Because every experimentalist, every working biologist has some questions that they think they understand, and modeling those is not terribly, it can be useful, but it's not really deeply useful. But they're the questions that are completely mysterious. And I would just like to say one of the, I think, most influential models we ever built, which is probably, I would say, half right and half wrong, was our first attempts to model what became known as homeostasis. In '93, Larry Abbott and I, with Gwendal Lemasson, published a model that was designed to address a problem which deeply, deeply confused me, which is how cells could manage to maintain approximately similar behavior over 60 or 80 years when channels were coming and going in the membrane in hours, days, and weeks. And so that very, very simple model, a very, very simple homeostasis model became, which is in its first instance, probably half right and half wrong, triggered a whole number of experimentalists, to us and others, to do experiments they never probably would have done that way if that model hadn't existed. And I think that's an example of, because I really, really didn't know how to think about this, and Larry immediately, after a year, figured out how to think about it. Today, we're doing a different kind of models, which is we're creating large classes of models to capture the variability in the populations that we study. And that is actually sort of more in what you're talking about, big data. It's sort of saying, well, how do we think about individual variability? And so I think, in what Christoph was saying, we sort of span both sides, but in all cases, I insist, and this is me personally, that all the models we build that can be quite simple or a little more complicated, that they don't transgress some of the true rules of what I consider how neurons work. So, we compute in conductance, and cells have thresholds, and I refuse to leave computing conductance and cells with thresholds. I can, what? And
Yes.
other people, but that to me is the golden rule.
So one thing to also, in this issue of like how much data do you need in a particular area to sort of productively model, I do think there is a lot of benefit to having a pretty decent foundation of empirical data to constrain the models, especially if you are interested more in a neuroscience based model, obviously. And so for my first modeling experience in grad school was in the context of the hippocampus, and there was a lot of data there over many years that people had kind of pretty well understood, but there was still enough opportunity to make kind of new contributions that you could actually lead to new insights with the computational model. So obviously you don't there is that sense of when a field is ripe, where there's a lot of data but not a lot of models, perhaps, or maybe the models that exist have some areas where they can obviously be improved. Those are pretty basic points, but I do think that is a pretty reasonable place to think about where modeling can be useful.
So I would like to mention two models that were important for me. The first is David Marr, not a neural model, but a role model. David Marr did absolutely splendid work, especially his early work when he was essentially a student, when he was 24, 25. And I learned about this when I was already older and he was dead. And this combination of things, and the fact that really not many people had really paid close attention to this early work. Then Marr became more famous for his later work just before he died. And Christoph, of course, knows a lot about it. But his early work, for me, especially the one on the hippocampus, was absolutely inspiring. And the fact that it was done by such a young person who had died was an irresistible combination of things. And the second model, in a sense, was similar, although the work is very different. Elizabeth Garner, who was a brilliant physicist who came into this community of physicists that wanted to learn about the brain. And she had this intuition that completely upended the approach in a way that maybe doesn't make sense to many people that now do computational neuroscience, because it was at a very abstract level, looking at an associative memory in the space of weights rather than in the space of activities. But it was something that was so innovative, it seemed, at the time. And she also died, unfortunately, early at age 31. I had the privilege of meeting her in Jerusalem, and then she died soon after. And for me, that was another inspiration of how you can do curiosity-driven research by just following your inspiration relatively, without caring about what others think about it, and without thinking about the impact it would have. Both David Marr and Elizabeth Garner took time and effort for others to realize what they had done. So for me, they were very inspirational.
Great. So I'm a little bit worried about the answer to my next question, because it seems like Alessandro's views mean that the person who works on the next big modeling area is going to die young. But I am curious if there are areas that you see now in the research landscape that seem like, modeling is being underutilized and they are ripe for it. So I guess for Eve, this would be an area where experimentalists are confused. It could be an area where there's a lot of data and people haven't tackled it yet. But are there research questions where you feel like, people should really be flooding in there with some modeling approaches?
Can I mention one that's one of the best explored brain areas bar none, which is primary visual cortex, which has been studied for 80 years. We have our models of it are incredibly primitive. There are variants of Hubel and Wiesel feed forward. they utterly fail to quantitatively describe, if you do what we do, which is sample 100,000 neurons in that, these models fail utterly, completely. They don't even approximate. They explain 2% of the variance, and people, have now all these deep convolutional these deep networks, because that's so successful in machine learning. But they have, of course, they don't at all resemble the structure of the brain, which is massively feedback. And so this really demands to be, it's a well-tended piece of brains, one of the best, understood one at some level, but we have no idea what it is actually doing. As was pointed out, of course, almost 20 years ago in a very popular review by Bruno Olshausen and David Field, what are the other 90% plus of cells doing in visual cortex? And if we don't understand visual cortex, how can we claim to understand, much of any other area outside maybe the retina?
So
one thing, just to jump in on that V1 thing is like, what does it mean to understand from a modeling perspective? And I think that's an aspect of the field that is evolving. But, I think the traditional sense of understand from a neuroscience perspective is I can literally see what a neuron's kind of receptive field or response property is and kind of intuitively connect that to a function, right? So like the classic example of a place cell or the Hubel and Wiesel kind of, edge detectors. But I do think, then there's, the nice kind of position statement from Richards et al. That is along these lines that basically is saying, hey, the brain is actually a learning system that is, adapting this highly parameterized neural network. And you may not understand in some kind of sense that you're going to really intuitively sort of directly see what it's doing. So I wonder, Christoph, if your characterization of V1 as being not being understood, what criteria are you using to understand when you say understand?
It's an excellent question. Being able to quantitatively understand or model how, their firing rate, their behavior, just trying to model it, right? We believe we understand the climate. We make all these predictions over climate. Well, here we take a piece of brain that should be vastly better understood because we can all study it in the lab. Can we predict how it's going to behave 10 milliseconds from now, 50 milliseconds from now, 100 milliseconds from now, and not at the very qualitative level, right? But actually at the quantitative level, that I can reproduce, some fraction of the variance of whatever variable I have. And if we can't do that, then our understanding, is very limited. Yeah, we can see we intuit it in some sense, but if you can't quantify it, what sort of scientific understanding is that?
I would just like to throw in, sorry, we have the second panel of today is a debate about this idea of can you understand computation or should you focus on understand learning? So if people want to dive more into that, that will be a debate panel later. Sorry, Eve, go ahead.
So I'm going to confess something, which since I'm old, I'm allowed to confess. And that is, I fundamentally don't even know how to think about large circuits. And nobody has done anything that has really helped me, although I think the experimentalists may start helping soon. And the reason I don't understand how to think about them is because in the tradition of people working on small circuits, there were some key parts of the problem. One is you had to be able to identify your neurons, so you could record from the same neuron over and over again in different animals. And two, you had to come up with a connectivity diagram. But you couldn't come up with a connectivity diagram until you had neuronal identification. And so where for years I always got stuck with large circuits was those rules no longer or were not in existence. So today we have much, much better experimental tools for identifying cells, or at least cell classes, and we're coming much closer to having connectivity diagrams or wiring diagrams with the connectomes, although those are still quite incomplete in some very important ways. But I still have a lot of trouble knowing how to think about how each individual cell is contributing to the whole dynamic of the network. And so I'm still stuck on thinking about that. And so since I'm still stuck on how to think about that, I still work on small circuits because at least I understand what the task is. But I can imagine in 10 years, we'll have tools that will allow us to get to an analogous place because of the molecular tools and the recording tools. That said, the big question is to what extent individual neurons up there in the real brain, our brains, have anything like specific roles, the way an individual cell in my circuit has a specific role. I don't know how to think about that question because I could imagine either way that there are sort of individualized roles that we just don't know how to even think about yet, or maybe that question just doesn't exist when you start talking about large circuits. So that's a question that I would hope that in 10 years we'll know how to begin thinking about, because I'm sort of open. And, I keep asking. And I'd just like to say one more thing. For years, people would tell me stories about how visual cortex worked. And they'd say, oh, it all requires, 1 million cells or, 10 million cells. And I'd say, well, what about how it works requires 10 million cells? And they'd end up telling me a 3-cell circuit description of how visual cortex or hippocampus works. I am waiting for an understanding that in any way, really, that people tell me in other than a small circuit way, because they're not there, I don't think yet, in a way that I can understand.
I mean, the bad news is that we now have the connectivity diagram of a cubic millimeter of primary visual cortex. We have the description of the molecular description of the 100 different cell types. We can record from, a large fraction of the neurons in this piece of brain in an animal where we control its input to a certain extent, but we still have great challenges. And I don't think in 10 years we will have overcome this.
And we had that information in 1980 on my systematic gastric nervous system, and we're still finding things out. So we've had that level of information for 40 years, and we still don't fully understand a lot of the fundamental biology. So in another 40 years, we can talk.
Well, it is this question of like, what is your criterion, right? So
predicting.
Me, I'm happy. Well, predicting is, but are you predicting like a particular brain, or are you predicting in general what you're going to see kind of as overall patterns of similarity? And one of the big things is, similarity is now, I think, a big coin of the realm where it wasn't before. And there is like, a lot of work that is basically focusing on representational similarity at different levels in the brain. And when you use that as a tool, it's obviously simplifying things drastically, but there is a good reason to believe that similarity is extremely important at a pattern level in the brain. And so if you just sort of say, okay, look, I'm going to focus on this kind of, level, very high level of understanding what's going on in brain areas, and say, yeah, I'm missing a lot of details here, but nevertheless, looking at how different brain areas sort of transform the patterns of similarity from, a raw visual input into something that you see in, infratemporal cortex at that level. And somebody put a link into, the brain score kind of data and stuff like that. And I think there is, again, a lot of progress that people would say, oh, well, I think we're understanding how through these like deep neural networks, how similarity gets transformed over many layers in a network. So it's not a detailed explanation, but it is a predictive explanation in the sense that it's matching data, but it's not matching neuron by neuron data. You're abstracting away from that.
No, but it's not. But ultimately, look, just take climate science. Where would climate science would be? Well, all we have to do is similarity patterns of future climates. No, we want to be able to predict. How bad is it going to get on the planet? And same thing in the brain. I need to understand given the current state of a brain, either a general brain or a particular this brain in front of me, how is it going to evolve over the next whatever time I'm interested in, and then let's match it up against an experimental system, and can I predict it? And if I can, then my understanding is incomplete, I would submit. And maybe you're right, maybe with brains, because they're highly heterogeneous systems, very far away from equilibrium, etc., maybe we'll never get there.
They may be even more chaotic than the weather. Who knows? Yeah.
So
yes,
I feel like this conversation could go on for a long time, and it probably will happen in this second panel later. So I do want to keep the focus on the role of modeling. But yes, I want Alessandro to have a chance to respond to all of this first.
I want to respond by making a small advertisement, even at the cost of appearing disrespectful to Eve. This beautiful book that I hope you can read is Brains Through Time by Georg Stritter and Glenn Norcutt. I was just at an online reading club yesterday, and it's on the evolution of vertebrate brains. That's why it's mildly disrespectful of Eve.
But those are real brains.
Those are real brains, and it's a beautiful book. And it has a phenomenology that we should try to understand, that very few people try to understand. So Christoph was talking about the visual cortex. There is a clear structure there in the visual cortices of species we are particularly interested in that we don't understand. I agree with Christoph, but there is even more basic structure of isocortex in mammals, our basic layer plan of cortex that we don't understand. And there are structures in our brain that go back hundreds of millions of years. There is beautiful work by Stan Grillner and his group on the basal ganglia that show how the fundamental traits of the basal ganglia are present in lampreys, in very remote cousins of ours. And those are things that we can observe, not me, but people who study them. There are clear patterns there. There is structure, and we should try to understand the structure. So I think these are all kind of domains that kind of excite our curiosity. There are plenty of things that we don't understand
So
in the real brains.
Just one little comment about this, which is that one of the challenges that experimentalists have to face and that theorists have to be wary of is when you are studying something which is an idiosyncrasy of your particular system, and when you've discovered something which is a route to a general principle. And so I think the best experimentalists are very good at using their own intuition to know how to study the idiosyncrasies because they need to, but to be able to extract general principles from that. And I think that theorists have to be more disciplined about sort of requiring the experimental data that they look at and asking that question. To what extent do you think you're looking at something which is idiosyncratic to the spinal cord of a lamprey versus the spinal cord of a mouse? Or to what extent is there a really fundamental general principle? And I think comparative work is a way of asking that question, but very few people actually are disciplined enough to ask them, either experimentalists or theorists, to ask that question. And I think it's fundamental to making progress.
Agreed.
Great. So yeah, I want to move on to the next question, but it is a little bit related to some of the things we were talking about in the understanding of V1, because I want to know how you guys have seen the field of modeling and computational neuroscience evolve in your careers. It does seem like there are kind of fads where people get stuck in understanding a system a certain way or applying a certain tool to everything. When the physicists came marching in the 80s, there was a lot of mean field analysis and things that were really very directly inspired by those approaches. Now we have a lot of social neural networks again. And so I'm just wondering, have you guys seen, what kind of progress have you seen either in the methods or the way people are thinking about it or the way that experimentalists and computational people interact throughout your career?
Yeah, all of the above. All of the above. I mean, in some very obvious way, it's now a big field. 40 years ago, when many of us started, there were very, very few people that were doing modeling of any sort. Now it's a large field with its own conference, with many books and journals. So in that sense, it's become much more sophisticated.
So I would like to say something which some people may find very insulting, but I'll say it anyway. Taking the prerogative of being the old lady. And that is, I think scientists often behave like sheep, and they're very faddist, and they follow fads. And I think theorists are even more likely to do that than experimentalists because they can move faster. If you're running an experimental lab, there's some amount of inertia in how quickly you can change what you're doing. But if you're a theorist, you can change from 1 minute to 20 minutes later. You can decide you're going to study something new and different. So I guess what I find disconcerting is how quickly the whole community moves to a new approach without thoughtfully examining the pros and cons of that. And so what I find really, really disconcerting is how, in particular in the theory community, but in the experimental community, it's true as well, that you end up with 200 or 2,000 or 20 people fundamentally doing the same thing. And you've sort of left aside the 3 or 4 people who might be continuing to do something different or with a different perspective. And I think it's actually the weakest, something very unhappy. And I think theorists are fabulous because they can move so quickly, and that's also the Achilles heel of the field.
I completely agree with Eve, and even more than she does. And I think I would like to point at 2 factors that to me have had a negative impact. The grant system and the publishing industry, both of which reinforce the kind of herd behavior of scientists in general and of theorists in particular. The fact that you have to, when you write a grant, to kind of think of what you might think about in the next few years, takes away completely the kind of feeling of going into uncharted territory and using your intelligence to move around something of which you don't have the coordinate of yet. So and the publishing industry is terrible. Is the fact that people get judged by how much they can impress their reviewers, where in a different field you are considered bright when what you do cannot be understood by your colleagues, whereas in these fields that are more business-like, you need your colleagues to understand you and also to think that what you are doing they could not do, and it's maybe useful to what they do. So I think it's just terrible. These have been terrible developments for theoretical or computational neuroscience. And
machine learning. You think it's okay if people publish incomprehensible stuff that no one else understands? You think that's an acceptable standard?
I completely think that in my time people were sending out preprints, and then preprints would certify priority, but whether people would be esteemed on the basis of their preprint, but whether somebody finds it's acceptable for a commercial journal is a different matter. Yeah,
I have two comments that don't really exactly relate. One is that from my perspective, computer science is the worst for fads. So there may be some kind of thing like, a few years ago it was like SVMs, everything's in SVM, and now everything's a deep learning thing. And you see that pivot, like you're saying, Eve, very quickly especially in computer science. So I guess to the extent that theorists are sort of along that vector, then that does make sense in that respect, relative to people who do empirical work. But anyway, so that does seem to be true. I don't think it's, exclusively true, but it certainly is a trend. But the other thing I wanted to say is, from my perspective, I've been impressed how much experimentalists have adopted a lot of important theory relevant techniques and essentially are doing a lot of work that theorists might have done, a few years ago themselves. And so if you look at typical papers that are showing up in the top journals in neuroscience, there's a lot of heavy data analysis techniques there, if not, actual modeling. And that's impressive to me because it shows that people, even who aren't quote unquote modelers, are really thinking about what they're doing from an empirical perspective in a much deeper way, in a more systematic way. So I do think that there has been an impact, I guess, of modeling, at least at some level, on shaping how people think about what's going on in the brain, and sort of, so to speak, upping the game for everybody in that respect.
I would like to say that I think the enhanced ability of the experimental community to do quality statistics is a tremendous step forward. And I think the fact that there were some very smart people who started out as theorists who were part of bringing sort of better statistics to experimental neuroscience is absolutely a plus. That doesn't mean to say that I'm happy that all of my postdocs are convinced that if they don't have bona fides in machine learning, they'll never get a job. And they're all convinced of that. So whether it's true or not, I don't know, but it doesn't matter because they're convinced of it. So they're just as convinced of that, and that's in 2021. They weren't convinced of that in 2019, and maybe in 2023 they won't be convinced of it, but they'll be convinced of something else. And so, I don't know how long these fads will last. Now, obviously, machine learning brings a lot of tools to people for many questions, but if every single postdoc is convinced that he or she has to have bona fides in machine learning, what does that tell us about herds and how we're training our people?
Yeah, well, my favorite metaphor about academics is basically cats rather than sheep. I mean, my perspective is that
cats are more independent.
From many perspectives, science is way more individualistic, way more diverse, and way more kind of everybody going off and doing their own thing and not working together and not collaborating, relative to many fields. I think that is true, and so I do think there are different perspectives on that science still has that ability to sustain lots of people searching their own gradient, so to speak, of the problem, and sort of in some sense actually avoiding what other people are doing so that they can carve off their own unique little perspective on things. So I think there are kind of both. I can see both sides.
I would say that's in your generation, but I'm not so convinced that our
the new generation is not that way. That may be true.
No, there's still tremendous. I mean, look, we lived through one of the golden ages of neuroscience, right? And one of the golden ages of computer science, right? There are a lot of very smart people. Yes, there's like in any other field of human endeavor, of course, there are fads and trends, but there's still huge diversity. You can find all sorts of interesting, crazy models out there. There's a huge diversity of funding, right? It's not just NIH federal. There's private funding, there's VC funding. So for any idea that's really good, you can work on it and you can get funding for it. I find it's a golden age. It's a fantastic time to be alive and think about the brain. We have never had it so good. Never before have so many people thought so hard about the brain. And I think that's fantastic, that we live in a society that enables us to do these things.
So you guys were mentioning kind of the next generation and what they're focusing on that. And so I do want to ask, before we get into audience questions, I do want to know what you guys think is the right kind of education for people who will go into doing some sort of modeling in neuroscience to have. Historically, people have come from very disparate backgrounds, even doing whole PhDs in physics or physiology before they come to any kind of modeling work. But now it's possible, even from the undergraduate level, to start doing computational neuroscience and be trained in that. So do you guys see a benefit of different styles of education, and what skills do you think people should really be focusing on gaining if they want to do this kind of work?
Get a quantitative undergraduate degree and then go work in a lab. Because if you haven't worked in a lab, if you've never confronted the reality of the brain, you are just missing some essential component of brains. So you want to have that, you need quantitative tools in physics and applied math engineering, machine learning, but then you have to spend some time probing any one neurobiological system.
So I would agree that it's very important that people learn fundamental basic principles of physics and math, etc., as young as possible. But to me, the most important thing to instill in every student is that they're going to be, if they're going to become a scientist, they have to be deeply understand the fact that they are going to be learning new things for the next 60 years. That you can't say I studied X when I was an undergraduate, or I did X as a postdoc. You have to say the world is changing and I'm committed to learning whatever I have to learn to go to the next step, because it's unpredictable what tools you're going to need.
I would just add, do things different from those around you. Diversity is the most precious asset in your education, our education.
Amen.
Okay. Okay, so let's go to some audience questions. People have been adding them in and upvoting. So I'm going to start with the one that has the most upvotes by far, and it's related to what a lot of the stuff we've been talking about. So what is the panel's view on the increasing use of black box deep learning systems as models of biological systems? What do we stand to learn from these systems that are powerful but have no correspondence to mechanistic constraints? I might take issue with no, but obviously very few correspondence to mechanistic constraints than traditional modeling.
Well, they help us understand one way how a particular goal like intelligence can be achieved, right? We've now seen we can get a machine that behaves in a certain situation, like playing Go or whatever. A protein folding behaves in a way that replicates some aspect, but that doesn't necessarily tell us that's the way how neuroscience does it or how neurobiology. But it puts strong constraints on our understanding. So I think I'm all for it, although they often do not replicate what actually goes on in the brain, but they're wonderful tools.
Yeah, and I think it's really important, it's kind of obvious, but, there's no one right model, and every model has trade-offs, and you have to look at things at different levels. And so, really you have to be looking at a whole stack of understanding at a kind of modeling level, from very low level detailed models up to high level kind of functional models. And in that context, understanding the connections between those levels is the most important thing, so that you can sort of build a connected understanding from those low levels up to the high levels. And that's kind of been the theme of my work. And we're talking about conductance-based models. I use conductance-based models, but my models also learn through error-driven learning. And so I feel like, one of the things I'm trying to do is try to make that bridge between like lower level aspects of the brain and higher level kind of learning principles that are at work in these kind of deep neural networks. And so to the extent that approach might be successful, that could be a way to say, okay, well, here's how you can translate some of the stuff about the high level functional results from the black box models and sort of see how that might then connect at a more mechanistic level down to ion channels and things like that. And just as a little kind of minor point here, that the NMDA and the GABA B channels turned out to be absolutely critical to get our models to do error-driven learning with spikes. So we're actually looking at those kinds of questions, and there are these kinds of potential, very detailed things may make a big difference at a biological level. So, all is not lost.
Okay, so we have time for a few more questions. I'm actually going to skip the next upvoted question because I think we talked about it a lot, about this concept of compact understanding of V1. So now the next question is kind of related to the perennial problems of building models, about kind of the level of detail that you put in. So Scott Rich asks, I find one of the biggest questions of modeling and presenting models to an interdisciplinary scientific audience is choosing where the line is between biophysical accuracy, i.e. matching data, and computational tractability, i.e. ability to reasonably simulate or mathematically analyze the system. In your experiences, how have you approached this challenge? Do you find that there's a general solution, or is it specific to the hypothesis studied in a particular work?
I think you have to
You got to
know
be pragmatic. It depends on the problem and the scale you're interested in. You've got to be completely ruthless, pragmatic, operational about it.
you have to really frame the question that you're interested in studying, and then make sure that you have enough detail to inform that question and not so much detail that you can't find the question. I mean, I think that's the key. I mean, you really want to make sure that you don't disappear the question, or that you, by going either with too much detail or not enough detail. So that's where theorist's intuition is so important.
One metric is like, what is the model going to tell you, right?
Right.
And that's kind of a guideline, right? So if you need to be talking about certain details that you know about, and then by putting that in your model, you can sort of understand something that you didn't understand before. That's the goal, right? And so that the answer to that is going to be very different for every different question that you're asking. But if you're not getting the model to tell you something, then maybe you're putting in too many details and the model's not kind of doing enough work for you, perhaps. I don't know. There might be some situation like that. I don't know.
And this goes back to the whole issue of general principles versus idiosyncrasies. And, I think there are lots and lots of models that were built trying to explain the idiosyncrasy and therefore losing the general principles, and others that, were just so abstract that you couldn't come back to the way biological systems work.
So that
the most impressive model to my, and the right level of resolution, if you go back to Hodgkin and Huxley 1951, when they had the rate model, and they hand cranked for 3 weeks, and to predict the propagation velocity, right, of the action potential, and they got it within 10%. That is freaking amazing. They chose the right level of resolution, right? They didn't even know about ionic channel at the time of it was microscopic current and conductances, and they wanted to compute this one physical thing, the propagation velocity, and they got it within 10% on a hand crank computer. I mean, that is freaking amazing.
But
for fourth order equations, right? Yeah.
But that's because they had a phenomenal deep understanding of biological systems, and they were quantitative, very, very, very brilliant. And so that is, that was just genius at work. And we all should try to have so much genius, right?
So the next question is related to this idea you were talking about, Eve, of idiosyncratic versus general principles. And Diana wants to know, what are your thoughts on how the current publication system allows for or doesn't allow for a clear distinction or discrimination between idiosyncratic findings versus general principles?
I think it depends on how carefully and how intelligently you write a paper. I mean, I think, and how intelligently And I will tell you one thing about in my own life, because I work on a very strange system, I have had for the last almost 50 years, had to always be able to explain to someone what I've learned that could be of general interest to them. And so I learned very early on to have the discipline to be able to distinguish between those two things. And if you learn that, then you always step back and say, why should someone working in the hippocampus care about what we've done? And sometimes I look at a paper and I say, I don't think the people in cortex are going to care about this result. And sometimes I look at the result and I say, I think everybody in neuroscience should care about this. So I think it's forcing yourself to be honest. I mean, I guess the thing is, I try to always force myself to be honest with how I think the results should be framed. There are papers I will send to, modest journals because I don't see the general principle there. I see it's something we have to publish because we have to publish it, but it's not going to be important to someone in hippocampus. And other things that I think will be important to people, even to Christoph, if he would read it. That was a little tweak, which took him a little while to get.
Okay, well, I think we are basically at the end of our time here. It was really great to hear from you all of your wisdom. I have to say, for me, the most surprising thing to hear was that the area that needs more modeling is V1. That's not where I thought we'd end up, but that's the point of bringing you all together for these discussions. So thank you so much to the panelists and to the audience for participating. They can all stick around, and there will be a series of short talks coming up next. So thank you so much.
Thanks
Bye,
a lot.
guys.
Thanks a lot, everyone.
Yeah, it was fun. Yeah, thank you.